The overall goal of this protocol is to quantitatively assess the effect of the high-salinity environments and transmembrane solution asymmetry found in physiological conditions on membrane phase states.
Solutions inside cell and outside cells are very different. Our methods help understand the effect of the solution asymmetry on the membrane properties and phase behavior.
The main advantage of our approach is that vesicles can be prepared at physiological buffer conditions, and the microfluidic device can be used to create asymmetric buffer conditions.
To begin the procedure, roughen one side of a PTFE plate with fine sandpaper. Wash the plate, a 15 milliliter glass vial, and a sealable glass container with commercial dishwashing detergent, ethanol, and chloroform. Use a 25 microliter glass syringe to deposit 10 to 15 microliters of lipid stock in chloroform onto the rough side of the clean, dry PTFE plate. Spread the lipid stock with the syringe needle to create a uniform lipid film. Use tweezers to transfer the plate to the 15 milliliter vial. Heat the plate at 60 degrees Celsius and desiccate for 2 hours to evaporate the solvent. Then, add deionized water to a glass container to a height of about 1 centimeter. Set the vial with the plate in the container, ensuring that it remains stable and upright, and seal the container. Allow the lipid bilayer to pre-swell at 60 degrees Celsius for 4 hours. Then, remove the vial from the container. Draw 5 milliliters of swelling solution into a plastic syringe and attach a 0.45 micrometer filter to the syringe tip.